Hi there guys, Mr. Lawton here. Very excited about this. This is going to be my first ever video showing you how to work your way through a Unit 3 past paper. So today's past paper is going to be June 2014. It's the higher tier paper. I'm going to focus just on B3 for this video, and then I'll do another one for C3 and another one for P3. So you get all three of the sciences and we just nail it out. The reason I'm so excited about this is because this is going to be the game changer. This is going to turn that measly D grade into the fantastic C grade that you deserve, or even higher. So it's going to turn your A's into A stars, B's into A's, and yeah, it's going to be awesome, okay? So watch me go through the past paper. I'll give you all the answers for it, and hopefully you're going to learn something from it, okay? So here we go. We go straight paper. Usual stuff, okay, the usual equations that we always have in all our past papers. Didn't mean to do that, well, so usual things. Most of them are for physics, but you have got a couple at the top that that you can use for chemistry as well. Not much for biology in here, okay, so on we go. Blank page, straight into it, okay, so questions about blood and circulation. Look at the diagram of a heart, and you just need to label the two parts of the heart. Okay, so remember, this is a higher tier paper, so we want as much detail as possible. First things first, you should be thinking, where is A pointing? Yes, it's pointing at a valve, but what valve is it pointing at? It is actually pointing at the tricuspid valve. Okay. Apologies for my handwriting, but it's quite difficult on the iPad, okay? So tricuspid valve, that's your 1 mark. Boom, done. On we go. Now remember that on your heart, we are looking at the heart as if it is in somebody's body, okay? So if it's in somebody's body, it's almost like left and right are reversed. So left is over here and right is over here. So we know that B is pointing at the left something. It's the first part of the heart that it comes into, and it is called the left atrium. Okay, so left atrium. Fantastic. 2 marks down, loads of marks to come. So let's move it down. The heart pumps blood out of the left and right ventricles. Explain why the left ventricle has a thicker muscle wall than the right ventricle. So again, you're thinking, why is this side of the heart, this muscle, thicker than this muscle over here? So you've got to think about it. Where do those parts of the heart pump to? You should be thinking the right ventricle has to pump blood a shorter distance, or you could say the left ventricle has to pump blood to the rest of the body. Okay, so that's 1 mark if you said left ventricle pumps to the body, okay? So the second mark is there if you are talking about pressure, okay? So if we're talking about the left ventricle is pumping blood to the rest of the body, we have to talk about the fact that there's more pressure, okay? So they need more pressure to pump all that blood to the rest of the body. The right side of the heart only pumps to the lungs, so there's less pressure. So you've got the inverse of those as well if you want to get those marks. So left ventricle pumps to the body, so there's more pressure. Smashed. First page done. Okay, here he is, a weightlifter, all right? So during a competition, he must hold the weight above his head with straight arms. This requires his muscles to use both aerobic and anaerobic respiration. Write down the balanced symbol equation for aerobic respiration. Now, whenever they ask you for a balanced symbol equation, there's always going to be 2 marks available. The first mark is for having the formula correct. Okay, so let's go for the formula correct. We should be thinking aerobic respiration. That means we've got glucose and oxygen being converted into carbon dioxide and water. So first off, we go glucose, C6, H12O6. Then we're going to have oxygen, which is our O2. I'm going to struggle for room here, but that's fine. Then we're converting it into carbon dioxide, CO2, and water, H2O. Just about fitted it on, fab. So that is 1 mark down. How are we going to get our second mark? We need to make this a balanced symbol equation. Now, with this one, I probably wouldn't even say that you need to try and balance it. You just need to remember what the balance is. And it's a really easy one. It's the devil equation, yeah? 6, 6, 6. That's it. Okay, 6 oxygen, 6 carbon dioxide, 6 waters. 2 marks, really easy marks, just from remembering a little bit there, guys, okay? So, here we go. Next part, for a successful lift, the weightlifter only needs to hold the weight above his head for 3 seconds. Weightlifters find it difficult to hold the weight in this position for longer than 3 seconds. Explain why, okay? So the main explanation points that we're thinking about, why is it going to be harder for them to get hold the weight over their head for 3 seconds? So our first point is going to be the idea that the muscles are not going to get enough oxygen, so not enough oxygen. Okay, so they're not getting enough oxygen, so what's going to happen? We're going to have anaerobic respiration happening. What builds up when we have anaerobic respiration? Then obviously we have lactic acid build up, and that's our second mark. Okay, so muscles, not enough oxygen, and it causes a lactic acid build-up in those muscles, which can obviously cause muscle fatigue and muscle pain, and so they're not going to be able to lift for as long. Okay, last part of this page, nearly done it. Okay, weightlifting can damage muscle cells, which need to be repaired. Muscle cells in weightlifters contain a large number of ribosomes. Explain why. Now, we don't need to spend loads of time on this because it is only 1 mark. So what is the one thing that ribosomes make? Then obviously you know ribosomes are needed to make protein. So you've got protein in there, then you pick up that single mark there. No need to spend loads of time on it. Ribosomes make protein. Done. Okay, on to the next page. This is easy, guys, yeah? So here we have it, 3 mark question, quite a meaty one. It's actually really easy though. There's loads of very, very easy easy marks that you can pick up on this one. So first off, let's read through it. We'll highlight some of the most important points. So pepsin and trypsin are enzymes, so we know that we're talking about enzymes, catalysts within the body, in the digestive system that break down proteins. Okay, in the stomach, the pH is between 1 and 2, very important. In the small intestine, the pH is between 7 and 8, fab. So now we're looking at the graph. It shows the rate of reaction of these enzymes when the pH is changed. Okay, so as you can see, pepsin's on the left, and you can see that it works in a range of, I don't know, between 0 and 3.5. We've got trypsin on the right that's working at a range of, say, 5.5 up to about 10, okay? So they work best at certain pHs, so we're thinking. Pepsin stops working when it reaches the small intestine. Trypsin will not work in the stomach, okay? Explain these 2 observations. 3 marks available here, guys. It's so straightforward, okay? So all we have to do is that we need to talk about that data. So first and foremost, we can say that pepsin does not work at pHs, so doesn't work at pHs of greater than 3.5. Yeah, we've got that on our graph. That's 1 mark. How easy is that? That is literally just reading data off of our graph over here. Yeah, that is all that is. So first mark, boom, done. We've got another 2 to hunt out, okay? So what we can also say is that the intestine's pH is outside of this range. So intestine pH is too high. Yeah, if it's too high, pepsin's going to stop working. We can see from the graph that if you go into the intestine pH, which is between 7 and 8 really, then it's going to stop working. The pepsin's going to stop. Fine. 2 marks. Happy days. Yeah, where are we going to get our last mark from? There's actually 2 really easy marking points again. So third mark is going to be doing the same for trypsin. So where does trypsin work best, or where does it not work? Okay, so you can see that trypsin doesn't work at pHs of less than 5.5, like I said earlier. Okay, 5.5. That's your 3 marks done. There's actually a 4th kind of marking criteria that you could put in. So if you said that the stomach pH is too low. There's 4 points there. There's literally no knowledge needed. It's all in that question. All right. So 3 really, really easy marks there that we should be answering. But I know there's some of you out there that would go, no, can't do it. Too much writing. Okay. So you're going to have to be the ones that are going to push this through and get those marks. Easy, easy marks. On this page. So, look at the rate of reaction for pepsin is also affected by temperature. Okay, so we've got another graph. They're killing you by graphs, but it's good because they're easy marks. They're giving us marks here. Okay, so we've got temperature along the bottom. We can see that it's moving up from 0 up to 70. We've also got the rate of reactions. Okay, so we just need to know that obviously the higher it is, the faster the reaction is going to go. So let's have a little look at the question. Calculate Q10 between 10 degrees and 20 degrees. Now, some people, again, are going to look at that. They're going to go, Q10, what the hell's that? I have no idea. I'm not going to even attempt this question. But you have to have a go. Please have a go at this, okay? It's not that hard. So you use the formula they've given you, rate at higher temperature divided by the rate at lower temperature. So it is as easy as just dividing two numbers. So we have a little look. Let's have a little look at the graph. It says at 20 degrees. So we go. You have to forgive my shaky hand now because there's not going to be a particularly straight line. But we go up from 20, okay? Then we come across from 20. Have a look at the rate of reaction. It's a 17, okay? Then we do exactly the same at 10. Go up and come across, and it's 8, all right? So those are our two numbers. We literally just do 17 because that's the higher temperature divided by 8, which will give us 2.1 if we round it to one decimal place. You can also get the mark for 2.13 if you've done it to 2, or 2.125 if you've got it to 3 decimal places. So easy, like these are easy marks, guys. Remember that C grade at higher tier is around about a third of the marks. If you're picking up all these easy ones, then you're going to do really, really well at this past paper. So the rate of reaction between 10 and 20 increases. Explain what the Q10 part in part 1 tells you about this increase. So it tells us, because it's a 2, that we know that it's the reaction is twice as fast. Okay, so reaction is times 2, twice as fast. Obviously, I'm writing just brief notes because I'm on the iPad and it's a bit tricky to write full answers. Obviously, don't write down what I'm, sorry, Mr. N. Don't write down what I'm writing. Write down in full sentences when you're answering in the paper. Okay. Next page, easy does it. Let's get it done. Right. So our next page is, here it is. The table below shows Q10 data for different enzymes between 40 and 50 degrees. Okay, so we've got enzymes, we've got this Q10 value. Enzyme A is most likely to come from bacteria that live in hot springs. So you need to use your knowledge of enzyme action to justify this statement, all right? So it's a bit of a tricky question, this one. Let's go back through it. So it's showing us the different enzymes between 40 and 50 degrees. So remember, the higher the Q10, the better they work at a high temperature, all right? So enzyme A is most likely to come from bacteria that live in a hot spring, okay? So enzyme A. Use your knowledge of enzyme action to justify this statement. Here we go. So the first marking point is for the fact that enzyme A is not denatured, okay? So remember, denaturing is when an enzyme, finish that. Denaturing is when an enzyme gets too hot and it changes the shape of the active site. So that active site, the lock and key active site that it goes into to get broken down, it changes its shape so it can't work anymore. But obviously, as you can see, because enzyme A has got a Q10 of 2, so we know that it's working twice as well at 50 degrees than it is at 40 degrees. It's a hotter temperature. It's not getting denatured yet, okay? So the second mark is talking about that active site, okay? So enzyme A is still working because the shape of the active site has not changed. All right? The shape of the active site has not changed. So that means that the optimum temperature for the enzyme is, or it must be, higher than the other ones, because the other ones are slowing down their reaction time. Ours is getting even better. Right, that finishes up the biology section. There is, of course, the dreaded 6-mark question, which we will go through in a separate video. I hope you found it useful. I will now spend some more of my time doing the physics and the chemistry sections, just so that you've got a complete past paper. Please look them up. Please have a look at them. Anything you don't understand, come see me, and we can sort it out. Okay. Thanks for listening, guys. Take it easy. Stay classy.